Thank you guys very much. So I guess my mandate over the course of the next 20 minutes or so is to pivot the conversation a little bit towards soluble factors in the blood and trying to enhance your enthusiasm that this might provide complementary information, really highlighting all of the biology that we can get from this type of profiling. At the same time, while I'll be very enthusiastic about this, I hope to not oversell what I'm going to be showing, because I don't want her as a roommate here, actually. Okay, enough background. So the overall goal of our laboratory is to identify associations, if you will, in humans, and then to go back to model systems to test for causal relationships. That said, we're, of course, very interested in biomarker discovery for sure, and a lot of my own thinking is influenced by my partner, Thomas Wang's work, who a number of years ago did some statistical modeling in collaboration with Mike Pensina, thinking about what it would take to identify new markers and how they might add to established risk factors and how that might modulate C statistics, a notion that Adrian and many others discussed yesterday. And as his starting point, he thought about some disease. Let's say it's incident diabetes, for which we have some good markers, baseline fasting glucose, BMI, et cetera. And what would we need to kind of budge the C statistic? And what you can see from this very nice modeling is that if the additional markers were fairly well correlated, say an R of 0.4, you'd need lots of markers and you would eventually sort of asymptote, if you will. By contrast, if you identify new markers in the blood, if you found things that were quite orthogonal to anything other information that you already had clinically, well, then that might be particularly fruitful in terms of disease prediction. And I'll kind of return to this theme over and over throughout the talk. Okay, so we were very fortunate a number of years ago to start with Tommy and Vasan Ramachandran and Marty Larson and others to begin to apply several technologies to the blood to look at disease predictors in the Framingham Heart Study. Okay, and we used several methods that use mass spectrometry, and in total we could look at about 500 at that time, 500 metabolites in the blood in a targeted fashion. Okay, so what I mean by that is you purchase a compound, you kind of establish its characteristics on a mass spec, and then you can multiplex, if you will, hundreds and hundreds of assays in as little as 50 microliters of blood. So we started off with blood from the Framingham Heart Study from this time point and looked at that time at incident diabetes 12 years later to see if there was anything biochemically in the blood that presaged the onset of disease. Okay, and here's the platform here, and I've just cut out all the known pathways from what's known as the KEGG database, but this gives you a broad feeling of what we can do now. The red dots are things that we can assay easily in the blood, and as I mentioned, in total, we measure about 500 metabolites quickly in a number of screens, but if you add the number of screens, as you can imagine, different chemical compounds have different biophysical characteristics. As you add more columns, you can assay now readily up to 1,000, 1,200, but we do about 500 in our quickest screens. Outstanding sensitivity, very good reproducibility, and the beauty is you can do this in as little as 40 or 50 microliters of blood with a high throughput. Okay, and these were our first findings in which we quickly realized that at the top of the list in terms of biochemical aberrations in individuals who were destined to develop diabetes was this clumping of amino acids, isoleucine, leucine, sorry about that. The isoleucine, leucine, and valine are branch-chain amino acids. Phenylalanine and tyrosine are aromatic amino acids. And as compared to some of the genetic information we described yesterday, for any individual one of these, the hazards ratios per standard deviation increment, or this is actually in the top quartile, but even the hazards per standard deviation increment were far greater than that. Now, of course, with the gene, it's somewhere in the causal pathway. This could merely be an epiphenomenon. And of course, we're integrating genetic information in a variety of different ways to get at that important question. And importantly, even if you adjust it for all clinical factors, as well as we had the results of an oral glucose challenge, et cetera, and other factors, hemoglobin A1C, anything you can really think about, this was independent of those factors. Now, Patrick yesterday mentioned a 3-marker genetic risk score. We honestly just picked sort of any 3 of these. These happened to be most statistically significant, but really if you pick any branch chain and any aromatic, they're orthogonal enough with each other such that if you look across quartiles, you begin to see a 3-4-fold increased risk of developing diabetes. Again, even if you adjust for age, sex, BMI, fasting, glucose, parental history, diet, and in this case, you actually modulated the C statistic quite significantly. Okay, so those are assays that we're doing with in a targeted manner, and many, many groups are doing that. I know ERIC is doing those sorts of assays, as are many types of groups. The real issue, though, and one of the take-home messages I'd like to underscore today is that the good news is we're all finding the same things. The daunting news is that there's a whole heck of a lot out there. I wrote that there's more than 10,000 compounds. There's easily 10 to 100,000 compounds in the blood, and we're really at this stage only looking at the tip of the iceberg. So we began to use these new technologies where you can, instead of just profiling all the known things in an untargeted way, look across all of the masses and begin to quantify things. And this was really enabled by the most sort of the recent iteration of mass spectrometers. And we asked a simple question: if you use these and assay everything, how many peaks are in a given sample? How many peaks are in the majority of individuals? How correlated are they with each other? How many peaks will be associated with any given phenotype? And how many can we quickly identify by a database search? So we went back to Framingham, looked at about 1,000 individuals, and as you can see, these are sort of absolutely free-range humans. I mean, they're young, 40-year-old BMI of 26, and we were fortunate to leverage CT scans that had been performed in these individuals, and we just asked a simple question: could we look at peaks that were associated with hepatic fat being a marker of incident cardiometabolic disease? So this is sort of the take-home of one of the real take-homes of the talk and of what we've been doing, and that is you see about 5,000 peaks observed in greater than 80% of the individuals. Importantly, if you do data reduction, factor analysis, it really only reduces to about 1,000 factors. So there really is, if we can identify everything in the blood, there's a lot of orthogonal information that's there, okay? And the other key point is that there were hundreds and hundreds of peaks that were associated with any key metabolic trait, such as hepatic fat. So as you can see, at a mass of 202.1185, there's a very strong association, it turns out, with liver fat. I'm just showing you at the bottom, we had published that glutamate, and then I told you about those branch chain amino acids. They're all associated with hepatic fat as well, but they're way down on the list, okay? So there really is a lot of information there. Now we just have to go through the arduous task of identifying it. This association held true even if you adjust for all the other potential covariates. So what are you stuck with? You're stuck with a peak and its association with some phenotype, okay? So what do? You go to a database and you try to look it up. So we went to that database, and we went to a series of databases, and this is a shortened, abbreviated list, and there was a very talented chemist in my group named Jordan Morningstar who basically quit about halfway through because he couldn't identify what this peak was. So I had had an idea for a long time, and influenced by many of you in the room around human genetics, we began to consider, these peaks are just continuous variables. And I began to wonder, wow, could we do genome-wide association studies for these peaks? Because knowing what the enzymes are or solute carriers, if they're associated with the peak, it might inform what the compound really is. And lo and behold, we found variants in a series of enzymes associated with this particular peak, one known as XII, which was actually mentioned in Josh Denny's group, and we're already talking about collaborations around this. So we found an enzyme associated with the peak. Any questions there? So then what you do is you go back to the literature, you think about potential enzymatic reactions, and I'll just quickly go to the punch that AGXT2 can take asymmetric dimethylarginine and make a compound known as dimethylguanidinovaleric acid, for which there were 3 papers in the literature, but had not yet entered the database, okay? So we tried to purchase it or get it from a group in Germany. They did not. Unfortunately, we weren't able to collaborate with them. We ended up synthesizing it. These are just mass spec data showing you that and chromatography data showing that we were correct. Okay, so that compound had an exact mass match. Okay, so now we went back and we developed a targeted assay with DMGV, and these are just data from the Malmo diet and cancer study showing you that we've identified a new predictor of incident diabetes, even if you adjust for age, sex, BMI, and glucose, with about a 2-fold increased risk. So the real thing I want to highlight here is I do think metabolomics has a real ability to inform disease prediction, but particularly we're going to need to identify, though, what is often referred to as the dark matter of the human metabolome. The other thing I wanted to raise just very quickly, because some discussed it yesterday, as we've gone on, it's nice to have a predictor of disease. It's really nice if it triggers if there's some interaction with therapy. So we've been collaborating quite a bit with the Diabetes Prevention Program, looking for predictors of metformin or lifestyle response. This is from a classic paper in the New England Journal, Knowles and colleagues in 2002. And what I want to highlight is that we have a paper under revision, but many of the metabolites that predict response to therapy, though, are not the same ones that predict in the whole cohort. And I think that that's something important to think about moving forward. So those are small molecules. I have 5 minutes to discuss proteins. So how are we assaying proteins in the blood? We've been doing 2 targeted approaches. We've done a lot of mass spec work because of the tremendous dynamic range of compounds in the blood around proteins. It's very, very hard to do mass spec for proteins. With the metabolites, you crash out all the proteins, and then you study. There's a little bit less dynamic range. It's a little bit simpler to do. So we've been working with 2 different assays. I can go into detail during the questions. One is called SOMAS, was generated by Larry Gold. These are single-stranded DNA aptamers that can be multiplexed, and you can measure in the assay that we have in a robot, using a robot in our own lab. We measure about 1,300 proteins. The company measures about up to 5,000 now, and we're doing a number of studies with them as well. We also use another system called Olink, and I'm going to show you some data from that. These are chimeric antibodies that have these DNA tails, and you can do PCR off those tails and measure now about 12 or 1,300 proteins using that assay. And I've actually, because I get asked this question all the time, these are unpublished data, but what I'm showing you here are distributions of pairwise correlations, just like that sort of theoretical modeling that Tommy had done. And what I'm showing you here is that with either system, there's lots of things that are greater than 90% of the proteins have pairwise correlations of greater than 0.4. Many of them have really very little correlation with each other. Now, I wrote up one of the caveats, though, is how specific are you really looking at signal as opposed to noise, and we can discuss that. But in general, there's lots of orthogonality in these types of techniques. So again, going back to the very same samples, this is a volcano plot, and we're looking at predictors of incident diabetes. These are in red are things that are age, sex, and multivariably adjusted for the things I mentioned previously. In blue are dots that are associated with in just age and sex adjusted models. And you can see on the top left over there is adiponectin, p-value of 10 to the minus 26 associated with incident diabetes, about a 2-fold decrease, and there are other factors. What's very nice is that as we've done this exercise, we'll see leptin, it's somewhere down on the bottom right. When we went fishing, we saw lots of things that should be associated with incident diabetes, and of course, dozens and dozens, in fact, 150 proteins in age and sex adjusted analyses that are associated with incident disease. Okay. And now the question is, well, what happens if you add that type of information on top of the genome risk score. Okay, so this study was published about 4 years ago, and we're just going to look at metabolites, and this is an early gene risk score. This was, I believe, this was done with James Meigs, who many of you will recall did one of the early risk scores for diabetes, and showed really very little, if any, change in ROC curves and other metrics. This is now taking about 31 variants, I believe it was, and then looking at metabolites on top of them, not proteins. We haven't done that yet. So here's the risk score, 0.641. Here are amino acid metabolites that I showed you alone. Here are lipid metabolites. This is from a different paper from Eugene Rhee in our group. It was published in JCI. And these are all the metabolites. And then, of course, look at the clinical factors on their own. This is for diabetes, so that's a tough thing to beat, 0.856. But if you add the metabolites on top of them, I'll let you judge. You go from 0.856 to 0.874, and then if you add all the metabolites plus the gene risk score, you go to 0.888. Again, a high bar to start with, but at least one metric to begin to get us thinking about this. Okay. So I'm going to end in the last 2 minutes talking about how we can leverage this technique in other data sets to identify new disease markers. So this is, I've learned now, this is a Dubai plot. I guess it's the provenance is from Dr. Bustamante. These are looking at our proteins in the Framingham Heart Study, and you see lots of variants associated with lots of proteins. And so sitting at a meeting a number of years ago with my friend Tommy and Jonathan Mosley and Dan Roden, we kind of came up with the following idea. And what we thought is, we really had very strong associations of these metabolites and proteins, very strong genetic associations. And so we had thousands of samples, but not hundreds of thousands of samples. And everyone had a plasma sample in Framingham. Everyone in BioView has, or other eMERGE, and all the groups that you guys are working with have DNA for sure, and of course many of them have plasma as well. Profiling them is a rather arduous and expensive task. And also, there are issues, some issues, of course, with getting archived samples that have been left around from pathology for several days, for sure. So what we did is we developed genetic predictors for each protein, and then we probed them to do PheWASs in BioView. Okay, so this was, I think Tommy or Jonathan, they called this virtual proteomics, which I think is kind of a cool name. And of course, the only way to do this would be if the loci, or many of your loci, explain large percentages of circulating levels. So it was very gratifying. A number of investigators mentioned ApoE yesterday. So across the y-axis here are a bunch of ICD-9 codes. Across the x-axis are proteins for which we had very strong predictors, okay? So if you go fishing with ApoE predictors into a PheWAS, you see liver disease, lipid disorders, eye disorders, and dementia, all the stuff that you think you would. Now look over on the right side of this slide, von Willebrand factor, critical for blood clotting, variant strongly associated with von Willebrand factor. Well, you go fishing, and the phenotype is up here, and that's thrombosis, so it made a lot of sense, okay? So obviously, as cardiologists, we're interested in those things that are associated with atherosclerosis phenotypes. So we made risk scores for those, and we found lots of proteins for which there was putative evidence, or when I say proteins, a lot of genetic protein predictors for which there was a lot of evidence that they were associated with phenotypes. And indeed, they were. And we went on to measure on the right-hand side our data hazards ratios per standard deviation increment in the Malmo diet and cancer study. So that was a genetic in silico assay that I described. Here is actually measuring these. And for sure, many of them cross the line. But overall, the pattern is strikingly similar. And as Amit mentioned yesterday, this is kind of a neat way, or the real interesting thing here is, do we really need more cardiovascular biomarkers? I would argue yes, better ones, but I think for diseases for which we don't have very good markers. Okay, so in summary, there's considerable orthogonality in the information ascertained by presently available profiling platforms. I think that targeted metabolite profiling and proteomic profiling appears to add information on top of clinical risk factors and polygenic risk scores. I really think that non-targeted methods, though, hold particular hope. It's a long slog to identify those. We're actually just starting 5,000 GWASs for all the peaks we've ever identified. And we're looking for predictors of disease, and also that they may not necessarily be the ones that are best predictors of response to therapy, and that finally that this virtual approach might inform biomarker discovery. So thank you very much, and I'm happy to answer any questions.
I think we have time for a few questions. Yes. We'll start here in the back.
Okay. Ronnie, is there any racial difference about this association?
Fantastic question. We are just finished about 2,500 samples that we have in the Jackson Heart study, and we'll let you know. We're also collaborating with the ERIC group as well around some of the metabolites and proteins for which there's overlap. So far, it looks like there are rather striking differences. I have to say, though, that the issue that we're having is that a lot of these platforms have evolved over time, so we don't always have apples to apples and oranges to oranges comparisons, okay? So there are more things in the platform. We're working on it. And then how about gender differences as well? Striking.
What is the average heritability across your measures?
So we about the 0.4, 0.5 something.
But that's
about
that's
0.4, 0.5.
That's,
we actually publish. I'll send you the figures. It's in a cell metabolism paper, and I think that some of them are 0.8, 0.9. Some are very low, and you see the differences between the essential and the non-essential amino acids.
Yeah.
It kind of has it's really nice because a lot of these things map, as I showed you, to enzymes, the things that kind of make a lot of sense. Dr. Valley, who was my biochemistry teacher, he doesn't remember this, a number of years ago at Johns Hopkins University.
Are you going to show pictures?
No.
Good. Thank you very much.
And I've been thinking a lot about what he does.
I mean, this is interesting in terms of prediction of risk, but I would think that metabolomics would make a great contribution to understanding disease mechanisms and rational development of disease therapy.
Absolutely. And I mean, I tried to say that early on. I didn't show all the data, but really what we're really interested in taking these observations, looking at, so let's give the example of the amino acids, right? So a lot of people said, we've just identified this epiphenomenon not epiphenomenon, but something downstream of insulin resistance, right? It's just you're catching insulin resistance better than insulin itself is. Maybe it's peripheral insulin resistance. So there was a recent study out of the UK Biobank recently where they did a GWAS for those branch chain amino acids. And that mapped to an enzyme, ppm1K, which is important in branch chain amino acid metabolism. It activates branch chain keto acid dehydrogenase. So again, it maps to an enzyme. That variants, those same variants, are associated with diabetes. So that's what we're really doing, is triangulating that information, and then when we have that information, then we're going to model systems. So yes, that's really what we're trying to do.
Okay. Oh, I'm sorry, Amit?
I was really interested in the predictor of response to therapy, specifically with respect to lifestyle in the DPP. Like, what are the predictors of response to lifestyle, and then what was the, measure of what your response was?
So it was abrogating insulin. So we did. So the paper is. I'd be happy to share it with you. It's coming. It's being reviewed or re-reviewed right now. So there are many lipids. There are amino acids, and there are several organic acids. I have to say it's a lot of lipids that predict who's going to respond to therapy, but not classic lipids, right? They're lipids of varying carbon lengths, of very specific entities.
I'm going to suggest that in the interest of time, thank you very much for
Thank
your
you.
talk. That was excellent. And now we're going to move to Dr. Nancy Cox, who's going to be talking about expression data.